Hello, everyone. Welcome to the GO-BGC OCB webinar. My name is Adam Stower, and I'll be hosting today's webinar. This webinar series focuses on updating the community on science being done with BGC Argo floats. And we have two speakers today, Camila Serra-Pompei and Martí Galí, for this webinar's theme of comparing BGC Argo observations with models. But first, we have Takeshita, who will be giving us an update on the GO-BGC array. So before we start, if the chat is open, so if you have any questions, I encourage you to post them there, or you could save them towards the end and ask them through the video call. So I'll pass it along to you.
Great. Thanks, Adam, for hosting today's webinar, and thanks, Camila and Martí, for going to give great talks today. So we're very excited to hear that. Yeah, so I'm going to start off with an update for the GO-BGC program. This is kind of what we do in BGC Argo for. This is a repeat of every webinar, but just kind of setting the stage in case there are newcomers to this webinar, which is what we always hope. Yeah, so BGC Argo is a part of the One Argo vision. So the plan for One Argo is to increase the Argo array to 4,700 floats globally. 2,500 of those floats will be core floats, so temperature salinity to 2,000 meters. 1,200 of those would be deep floats, and 1,000 of them would be BGC floats doing missions to 2,000 meters and carrying biogeochemical sensors. And the justification behind the number of 1,000 BGC floats is outlined in the Biogeochemical Argo Science and Implementation Plan. We are actually in the process of updating this plan, as hopefully a updated version will be published in sometime next year. But the main aspects of the implementation plan haven't changed much. And then so the implementation plan calls for 1,000 biogeochemical floats equipped with oxygen, pH, nitrate, chlorophyll fluorescence, optical backscatter, which is a proxy for particles, and also downwelling irradiance. And that's kind of globally and evenly distributed throughout the world. Just as a reminder, I think most people know, but all BGC Argo data and all Argo data are made freely available, both in near real time and also as a delayed mode quality controlled data set. The main access point is the Argo Global Data Assembly Center. That's what GDAC stands for. And to access BGC data, we strongly encourage the use of SPROF files, which are synthetic profiles, and which just means that the pressure axes for all of the BGC and the core data are interpolated into a single pressure axis. So it just makes it a much more user-friendly data set to use rather than the individual BGC profiles. The GDAC now has monthly snapshots of the SPROF data. So if you want to write a paper or do analysis that's traceable, then we strongly encourage people to use these snapshots. So that's associated with DOI, so people can recreate your analyses. The group at PMEL has really led the development for the One Argo toolbox, which is available in MATLAB and R. And so this makes this a toolbox that is open source. You can access through GitHub. And it makes accessing and filtering through, choosing what profiles you want from the Argo dataset very straightforward. So people starting out, we strongly encourage you to try out the One Argo toolbox. For the GO-BGC and SOCOM projects, we also have additional data snapshots. So the floats deployed through these two programs, we go through a DMQC, so the delayed mode quality control process twice a year, and then archive that at the UCSD online library as a DOI traceable product as well. And the main difference between the GDAC and this is that we also provide derived carbon parameters, so PCO2, DIC, alkalinity, and POC from the floats. So if you want these products for the floats just from SOCOM and GO-BGC, you can access these through these twice yearly snapshots. And we have started a data FAQ page on our website, gobgc.org/data/data FAQ. We're trying to compile commonly asked questions about for example, data quality, accuracy for these sensors. And so if you have any suggestions or questions that you would like to have answered on the FAQ, please email me. Moving on, so this is what a hypothetical 1,000 float BGC float array would look like. This is just randomly placed dashes globally. And this is our current status with BGC Argo. This is so not only the US, but this is a global. And so there are 584 operational BGC floats right now, and 463 have at least 4+ sensors. And that's 43 more than the last time we had this webinar. And now there's 67 with all 6 sensors, which is 13 more than what we had in the last webinar. And as you can see that the BGC Argo array really has achieved a global coverage. And so there's a few holes, that I'll kind of go into in a little bit. But this data set really has reached a global array, so that really opens up new doors for analyses that we have been hearing through these webinar series, and hopefully we'll hear from today's speakers as well. Since the last webinar, we have the GO-BGC has deployed 36 floats, largely in these areas that I've highlighted in white. And just kind of a little celebration, we have now surpassed 10,000 GO-BGC profiles this July, and now we're actually up to about 12,000. So that's exciting. So that was a little bit of a celebration moment for this program. And we have, so GO-BGC was funded to deploy 500, so we have about 300 more to deploy in the next couple of years. And we have 100 float deployment plan over the next 6 months or so. So each one of these solid symbols represent a float that is to be deployed, and then the open circles are floats that were recently deployed. And then so we're trying to fill in the big holes. So in the Southeast Pacific, for example, in the Indian, this is a GeoTraces cruise. This is a I-9 North. This is A16 South. So we are trying to fill in these holes in the ocean basins to kind of get more even coverage. So these should be deployed in the next couple of months. So please keep an eye out for those deployments. The BGC effort is growing internationally as well. So this is a little bit outdated. We're actually having the Argo data management team meeting next week. So we'll have updated numbers. But as of last year or maybe 2 years ago, there were about 250 floats targeted from international partners, largely in these regions, but I'll have an update on updated numbers for those in the next webinar. From the perspective, a couple updates specific to GO-BGC. We are now deploying more and more 6-sensor floats. So we have now deployed 26 6-sensor floats. And those are coming from all 3 float types: the BGC Apex, the Seabird Navis Nautilus, which is the version 2 of their profiling floats, and also the BGC Solos, or the S2As. So we have 3 float types now that can carry all 6 parameters, and we've been deploying them around the world. And in particular, so the MRV BGC S2As are the newest, commercially available BGC floats. And so we have deployed 10 of these in the Pacific now. So that's the big circles, and they're all working great. So that's very good news for, for this program and also for BGC Argo. It really seems like these floats are operating well. So that's another commercially available BGC float that people can purchase. One point that I want to kind of bring home is that future GO-BGC and BGC Argo is still uncertain. There is no secured funding after the GO-BGC program ends in 2 years. And so a lot of people, just from conversations, I think people will assume that BGC Argo and this wonderful data set will continue into the future, but the future is really highly uncertain. And so I just kind of want to, just when you have conversations with people, I just kind of want to make sure that that point is accurately portrayed, because it's, if the community really wants it, we really have to hear from the community, everyone on this webinar and more, that this data set is valuable and it should be prioritized for funding. So a couple of things that, you all can do, concrete things, write proposals using BGC Argo data that shows to the funding agencies that this data set is valuable. Write papers using BGC Argo data. Obviously, that also highlights the scientific utility. And third, this is really important, please cite us properly So how to cite Argo data is outlined in the Argo website and also the GO-BGC website, and really helps us track who is using the data set. And so that's really important. So if you're reviewing papers, or if you're writing papers, please make sure that the Argo data and BGC Argo data are cited properly. And the final update I have is that the GO-BGC program is organizing the another science workshop, the BGC Argo Science Workshop in the summer of 2025. That's next year. And it's going to take place at the University of Washington, most likely. So this one, we're currently still, kind of deciding on the scope of the project, but the initial idea is that we're going to have a 4 to 5 day hackathon style workshop. So be really focused on, diving deep into the data and try to produce some science, kind of get scientific results from profiling flow data. One of the benefits of hosting it at UW is that we can tour the UW Float Lab. The last workshop, we got a lot of feedback saying that they wanted to, see floats. So we have listened. And again, another typo, it's going to most likely be in the summer of 2025. Once those dates are set, we will send out email to the community. So please keep an eye out for that. And with that, I will pass it on to Adam.
Okay, so for our speakers today, we have Camila, Serra-Pompei, and Martí, Galí. Our first speaker is Camila, who is currently a postdoctoral researcher at the Technical University of Denmark. Camila is interested in combining global biogeochemical models with field and satellite data to better understand plankton dynamics and marine ecosystems. And we'll be presenting a talk called Assessing the Potential of Backscattering as a Proxy for Phytoplankton Carbon Biomass. Our second speaker is Martí Galí, who is a senior researcher at the Institute of Marine Sciences in Barcelona, Spain. In the past, Martí has done work on biogeochemical cycling of dimethyl sulfide, or DMS, and related compounds produced by microbial plankton in the upper ocean, but has recently focused on the biological carbon pump and the cycling of detrital carbon in the ocean's twilight zone. Martí will be presenting a talk on called Constraining stocks and fluxes of particulate organic carbon through the comparison between particle backscattering measurements and the Pisces version 2 model. So without further ado, I'll pass it along to Camila.
Okay, can you hear me? Fine. Okay.
Yes.
Yeah, thanks, Adam, for the introduction. Yeah, even though the topic of this talk is gonna be, was supposed to be comparing models with BGC Argo, I will do that in one of the slides, but actually what I will do is to use a model to better understand a product that is derived, or that can be derived from BGC Argo backscattering data. And that is phytoplankton carbon. Yeah, so the problem is that phytoplankton carbon in the ocean is hard to measure, basically because it's very hard to separate cell by cell from the other cells in the ocean, like heterotrophic bacteria or zooplankton. So often what we use is a backscattering coefficient as a proxy for phytoplankton carbon. And this is often used as a linear regression, relating the backscattering at a given wavelength to the phytoplankton carbon. The problem is that since we have so few data, we cannot really know how well the backscattering coefficient is a proxy for phytoplankton carbon. And the reason is that backscattering is not a property unique to phytoplankton. It actually, like all the particles in the ocean, scatter. Heterotrophic bacteria, zooplankton, detritus, minerals. And not only that, but also the cell composition or the cell size of the organisms can affect the backscattering coefficient. So basically, in this project, what I will be doing, or in this talk, I have divided it into parts. In one of them, I kind of review some of the existing phytoplankton carbon algorithms based on backscattering. And in the second part, I will do this modeling exercise that will address the relationship between backscattering and phytoplankton carbon. So from the algorithms that we have that use field data of backscattering together with phytoplankton, we can see that there are quite a few samples for each study. And at least from the studies that I have found, they all use different methods to measure phytoplankton carbon. For example, they all use flow cytometry, but some of them, for example, in this one, well, mostly one of them, in the graph et al. 2015, they basically measure the samples and analyze for total carbon content of these flow cytometry taken samples. And then for the larger size classes, they assume the same carbon to chlorophyll ratio and estimate carbon based on chlorophyll for the larger size classes. On the other hand, the Martinez-Vicente basically centers only on pico and nanoplankton. So it's not fair, I would say, to compare with other algorithms that assume the entire size spectrum of phytoplankton. But still, they used also, for example, literature data of cell to mass conversions to obtain the carbon biomass. Finally, Q et al. 2021 basically uses also flow cytometry, but will assumes a size spectrum to derive the phytoplankton carbon of the larger phytoplankton size classes. So as you can see, all these algorithms that have been developed using field data cannot really be compared between them. Another thing is that to overcome the scarcity of these of field samples of phytoplankton carbon, chlorophyll can also be used. That's the case of this paper from Berenfeld et al. 2005, where they basically took, I think, satellite-derived chlorophyll and backscattering, and then they derived some kind of relationship, which, well, it's this one here. And this basically assumes some kind of community average chlorophyll to carbon ratio to obtain these coefficients. And yeah, and this is what I will be doing in the modeling exercise afterwards as well. I discuss more in the paper in the appendix if anyone is interested on the derivation of this. So yeah, we can also use chlorophyll basically. So I took all these algorithms that I could find and I put them all together. I converted them to the same wavelength as they all initially were measured at different wavelengths of backscattering. And this is what we can see. So at large values of the backscattering coefficient, they all converged, except the Martinez-Vicente, which is slightly different. They all converge into similar values. However, when we look at low values of the backscattering coefficient, you can see that all the algorithms differ quite a lot between each other. And then, yeah, you can start thinking, okay, which one is the best one? We don't know. I would like also to note that the graph et al. Is the only algorithm that has a positive intercept, which might not be ideal because that means that basically when there is absolutely no backscattering, there is still phytoplankton present. So yeah, I would expect the algorithms to have a negative intercept. But the question was, okay, but are we ever really within these backscattering values, or are we mostly within here and then it doesn't matter which algorithm we use? So to test that, I use BGC Argo flow data. And this is what I'm showing here. Basically, in these plots, I'm showing where there was Argo data of the upper 10 meters for different months. And then the blue dots show basically the data points that were below this threshold, the at least 3 times difference threshold between algorithms. And here, basically, I show the cumulative data for different regions and for different months of the BGC Argo flow data. And basically, what we can see is that actually, for example, here in this tropical region, about 60% of the data for most of the months, well, between 25 and 50, let's say, is below this threshold. So it might matter which kind of algorithm you might be using. And even, for example, during the winter, in some region, for example, here, or for example, in oligotrophic gyres during some months, you can even be below this very big threshold here. So yeah, it will matter which kind of algorithm you choose in those regions. However, yeah, after the study, I thought, okay, actually to convert this backscattering value, so the algorithms to the wavelength at 700, I assumed a gamma equal 1. So a spectral slope of backscattering equal 1. But then I remembered actually that this spectral slope can vary across regions. So I tested, okay, what if I assume a different spectral slope of the backscattering coefficient? And here I redid the same plot as before, but these new slopes, we get these new thresholds as here in red. And we can see that actually, if I assume a higher spectral slope, well, you will get less of the data below these thresholds. So good news for whoever is using ArgoFlow data. At least for the oligotrophic regions, there is less data below this threshold. But well, these are things to keep in mind. So basically, to conclude this part of the talk, we could say that we need to constrain better the algorithms, the phytoplankton carbon algorithms that we have within the lower backscattering values, which are important for oligotrophic gyres and especially for winter months in temperate and polar regions. Okay, so the second thing that I will be doing is to explain this modeling exercise that we did. Basically, what we did was to take a global biogeochemical model that has several functional types of phytoplankton, and also have several size classes of zooplankton and heterotrophic bacteria. So here you have basically the food web that we used. And as you can see, we have coccolithophores, diatotrophs, diatoms, mixotrophs, zooplankton. And basically in this model, we have assigned IOPs to all these organisms. And here you can see, for example, the backscattering parametrization for all the groups. So here we have the specific backscattering coefficient per particle, basically. And this with wavelength, and here with compared to the cell diameter for each functional group. And then we have a radiative transfer model that also allows to simulate light trajectories and the RRS signal in the surface. Anyway, we use this model to understand better how backscattering algorithms estimate phytoplankton carbon. But before that, we wanted to check if the model performed more or less well. And this is my slide comparing Argo with models. And basically here we have in the x-axis, we have chlorophyll from the Argo floats and this chlorophyll from the Darwin model or the GCM biogeochemical model. And here it's the same, the backscattering from Argo and backscattering from the model. And each marker is a region following Longhurst regions for different months. And the error bars are standard deviation simply of doing the mean of all the data in both cases. And as you can see, I mean, I would say the model performs relatively well, but in general, but we tend to underestimate quite a bit the tropical regions, and we tend to overestimate for the backscattering coefficient in when we have high values of backscattering for the temperate and subpolar and polar regions. But yeah, the model seems to perform reasonably well, so we are happy with that. And then what we did basically was to use a model as if we were doing in the field. So basically we take all the phytoplankton carbon data that we have here. Luckily, we know exactly which one is that value, and all the backscattering coefficients that we have for each bin of the model in the surface only. And so basically, we don't have any methodological uncertainties. Here, what I'm showing is a backscattering from the model and the phytoplankton carbon biomass from the model in the linear scale. And this is exactly the same, but plotted in the log scale. Okay. And then we generate our own algorithm, as the others have been doing, just by using a linear regression. Then I do the same here using the chlorophyll times this Q factor that I called, which was basically some kind of community average chlorophyll to carbon ratio. Okay. And as you can see, because I know which one is more or less the best value, we have basically almost the same values than directly the phytoplankton carbon. It's just that the spread is a bit wider. But all else, it seems to perform quite well. Of course, it assumes that you know more or less what is this Q factor. I also want to say that because most of the data center in the lower values of the backscattering and of the phytoplankton carbon, we had to use some kind of robust method for the linear regression, as the ordinary least squares was not really performing that well, because it was quite affected by the larger values. Okay, so if we look at the results of, or the performance of these algorithms on the model, here I'm basically showing the real phytoplankton carbon biomass in the model. And here is the bias between the phytoplankton carbon biomass derived using the algorithms from before. So in this column, we have the ones derived directly using the phytoplankton carbon with backscattering. And the ones on the right show the same, but derived using the chlorophyll to backscattering. So basically what you can see is that in general, the errors are, the mean absolute errors at the global scale are not that big. We are talking on errors on the order of 20 to 30% on average, but there are large regional differences. Especially, for example, in the Southern Ocean, we tend to overestimate, well, the algorithms tend to overestimate in winter and underestimate in summer. The same goes for other regions. But yeah, there is high variability. And again, there is not that much difference between whether we use chlorophyll or whether we use the phytoplankton carbon biomass. But again, in here, I do know what is the best coefficient to match these two values. So there is a bit of a trick. Next, well, basically what we can see is also try to identify what is the contribution of each one of all these components in of the total backscattering signal. So for example, here I'm showing the contribution, the percent contribution of phytoplankton to the total backscattering for 2 months of heterotrophic bacteria and of detrital particles. And as you can see, most of the signal is dominated by the detrital particles, but phytoplankton can still have a strong or large contribution of the backscattering signal at high latitudes in summer. And interestingly, heterotrophic bacteria can also have a relatively high contribution during the summer months at high latitudes, up to about 30%. And next, I wanted to see a bit how each one of these components affected the relationship that we see between the backscattering and the phytoplankton carbon biomass. So basically what I did here is to plot, so here in the y-axis, I have the log 10 of the phytoplankton carbon biomass in the model. And the x-axis, I basically plot here only the backscattering contribution by phytoplankton. So we have this direct relationship. Then here we have of the backscattering of all plankton together. So phytoplankton plus zooplankton plus bacteria. And finally, this is the shape that we end up getting, which was what I showed at the beginning, using all the particles in the model. So also including the detritus. So basically, I'm showing here that the intercept, the kind of the intercept of this relationship is set mostly by the detrital particles and not so much by photoacclimation, which was a discussion at some point in some of the articles. But of course, I'm only looking at surface data, so I haven't really looked at DCM, which might affect maybe this, but I'm not sure. Especially if we were taking into account the chlorophyll one. And here, basically, I did a simulation, but assuming that all phytoplankton scatter in the same way. So we don't find differences in size or by phytoplankton functional type. And then we can see that actually a lot of the variance that we can see in the relationship between total backscattering and phytoplankton is not mostly driven by phytoplankton functional types, but mostly actually by other plankton in the water and also detrital matter. So yeah, maybe implementing functional types of phytoplankton might improve the algorithms, but yeah, maybe it needs to be tested better. So yeah, that's about it for the talk. The concluding points are basically that there are large differences between existing algorithms at low backscattering values. So basically we need to try to find or better constrain these algorithms. We have provided some kind of minimum average error that can occur if we don't have any methodological uncertainties or sampling biases, which we have nowadays in the field. And these go between 20 and 30% at scale, but we have quite big regional differences. Yeah, as I just mentioned, phytoplankton functional types only explain part of the variability in the relationship. And yeah, as I showed earlier, most methods that we use to derive these algorithms use different methods. Actually, there is almost not a single paper that used the same method as any other paper. So it would be nice if we reach some kind of consensus on how to achieve this. And yeah, thank you very much. And I will be happy to answer any questions.
All right, thank you, Camila. And I think a lot of people are interested in that the phytoplankton carbon to backscattering relationship, and including myself, and understanding that uncertainty around it. So thank you for your talk. We'll keep test questions to the end still, but we'll move along to Martí. So.
Great. So let me share screen. Hi everyone, and well, thanks for having me today. And as Adam already introduced before, I'm talking about how to use BGC Argo observations, and in particular particulate backscattering, to better constrain the stocks and fluxes of POC in a model, in a biochemical model. So the framework of this talk is basically the data of the biological carbon pump, which, as you know, is the vertical transfer of carbon pools mediated by biology to the ocean interior. And we know historically that the focus has been on the mechanisms that regulate the vertical export from the euphotic or productive layer of the ocean. But nowadays, we know that we still know very little about what regulates how much of this exported carbon and how this exported carbon is attenuated, this flux is attenuated through the mesopelagic layer. So how much of it is respired, or how much of it is, for example, transferred to upper trophic levels. And essentially, this means that mesopelagic carbon budgets are very poorly constrained. And one classical way of studying this topic is using long-lived tracers like phosphate, oxygen, and assuming that the ocean circulation is at steady state, so that all the carbon that is exported and all the organic matter that is exported is then remineralized and must match the vertical supply of nutrients to the euphotic zone. This is a powerful approach, but it has limitations because now we know that we are in an ocean that is not by no means at steady state because of climate change and the changes it induces in circulation, in ecosystem dynamics. And then on top of that, we have interannual and multidecadal variability. We also know that carbon export events and the mesopelagic metabolism they trigger are very episodic in nature. And we still have very limited understanding of the export mechanism, but also of the mesopelagic POC budgets, as I was mentioning before. And all this translates into a simplistic representation of mesopelagic and POC and cycling in models. So several years ago already, at some point I set out to study this topic using BGC Argo data. And because I guess as many of you, I became fascinated with these data sets that show all these events over annual or interannual cycles, including here blooms at the surface, export events through the mesopelagic, and deep convection events in this particular region, where I have been focusing in the North Atlantic, Western subpolar gyre, and so on. So this is my working hypothesis for this work, which is still a work in progress, and is whether we can use these high-resolution backscattering profiles to improve the model representation of the carbon pump. Good. So this was my introduction, and now we get into the the problem. And the problem we have here is comparing, not just comparing a model and observations, but also getting them to talk to each other. And said in another way, optimizing a model against observations, right? And when we attempt to do this, we have to consider many sources of uncertainty from both observations and models. So for example, in the observations, we have uncertainty in the measurements, in the calibration factors, conversion factors when we use proxies, as for POC and backscattering. In the case of backscattering, we have the sampling uncertainties and representative undersampling uncertainties. In the model side, we have uncertainty in forcings, boundary conditions, also in parameter values, so the scalars which have fixed values, which represent rate constants or coefficients in model equations, and we have uncertainty in the same model structure, so the equations themselves. We also have another type of uncertainty, which relates to how we translate observed variables to simulated ones, which sometimes they have a clear equivalence, sometimes not so much, as you will see. Okay, so I will go back and forth from this scheme. And I will start talking a little bit about this uncertainty in the observations and the processing I applied for my project. So when we talk about the backscattering signal, as you know, in recent years, some colleagues have developed, like Nathan Briggs in particular, have developed these techniques to process the signal in a way that we can separate a smooth baseline signal, which corresponds to the bulk of the particle assemblage, and the spikes, which correspond to large individual particles passing in front of the sensor window. So applying this processing, we can separate small POC, which is mostly suspended or slow sinking, and large POC, which corresponds mostly to the fast sinking fractions. Okay, now we have these two pools from backscattering, and we want to convert them to POC to enable this comparison to the model. We could do this the other way around and convert and get the model to simulate backscattering, as Camila did, which I think is a very wise option in perspective. But this is what we did here. So I realized that we didn't have an appropriate approach for this conversion. And basically in this paper published in 2022, I proposed this very simplistic approach where we have a maximum POC over backscattering ratio that is maximum at the surface and decreases following a power law function as proposed in previous works. And I use different surface conversion factors for different ocean biomes. And implicit in this simplistic scheme is that it is plankton ecosystem dynamics, and grossly speaking, that drives these variations at the surface. And it is detritus transformations and the progressive loss of organic carbon, and more rapidly than the mineral component, for example, what drives the decrease in POC over backscattering with depth. And well, this is super simplistic, but in a very nice paper that was published recently, they showed that this approach was doing relatively well. And you can see this Galí 22 here in the middle when compared to a more sophisticated approach that takes into consideration both backscattering and fluorescence to predict POC. So taking into account also the algal or fresh organic matter component. I recommend also this paper. Good. So now we have the observations. We have backscattering profiles converted to POC pools. So the next step is looking at how this corresponds to simulated variables in my model. And first, I have to describe a little bit the model I have been using, which is the PSCES version 2 model, which has been used for CMIP 5 and CMIP 6 simulations, but it is relatively complex compared to most CMIP class models. So essentially, this model that you can see here in this schematic, it has two classes of detrital pods, small, which sinks slowly, and large, which sinks fast. It has several pathways that lead to detritus production, including mortalities and assimilated ingestion or sloppy feeding, physical coagulation, and so on. All of this is explicit in model equations. And it has two particular features that are not in other models. One is it represents mesozooplankton flux feeders, which intercept the rain of sinking particles and aggregates and fragment part of the large aggregates into small ones. And it also has an important feature, which is that the degradation of POC, it uses a formalism, a mathematical formalism that is called the reactivity continuum, which allows, so without increasing the number of POC tracers, it implicitly represents the POC as a mixture of labile POC, which is lost very rapidly in the water column, and refractory POC, which can be exported deeper. Okay, so how do we compare this to observations? Theoretically, as you can see in this table, the small POC from backscattering should correspond to the two phytoplankton types in PSCES, microzooplankton and small detritus. The model also has eutrophic bacteria, but not as a tracer, so it's only implicit in the equation, so we are not considering it here, although it contributes, of course, to the observed backscattering. But it could be also kind of lumped together in the POC in the detrital fraction in observations. Anyway, the large POC corresponds theoretically to large detritus, which are over 100 microns, and mesozooplankton. So this is our assumption. And what we did is, first of all, seeing in 1D simulations how an annual cycle of BGC Argo, which you see here on the top left, compares to the simulations in PSCES, which you see on the top right. So to do that, we basically took the vertical mixing that resembled most the observations, which is not easy to see in this plot, sorry for that. And we see that the model captures more or less the magnitude of the POC over the water column and months, although it has some mismatch patterns. So we then generalized this to several annual time series from different floats all over the globe. And here you can see a summary for different ocean biomes. So on the top, you see the epipelagic layer, and the y-axis is the PSCES POC stock, small POC, and the y-axis is the BGC Argo observations. So this is the 1-1 perfect agreement line. And as you can see, in most biomes, we see a reasonable good match in terms of a small POC, and which was most surprising also in terms of the large POC. So you see an important correlation, which is the order of R squared of over 0.6, I think. Although the PSCES model tends to overestimate this stock according to the calculations we made, which are uncertain themselves. In the mesopelagic, the model skill or the comparison between model and observations is a bit worse. Okay, so we also compared the model and observations in 3D. And so looking at the seasonal profiles in each biome and subsampling the model at the places where we had observations, which you see here in the very small dots, black dots. So I'm not going to show these profiles directly, but I'm showing this comparison between the skill of the 1D approach and the 3D approach. And this would be very long to explain, but basically the large symbols correspond to the 3D model and the small ones to the 1D simulations tracking BGC Argo floats. So here you see the correlation on the top. In the middle, you see the ratio between model and observations, which should be 1 ideally. And on the bottom, you see the bias. So in general, we observe that the 1D approach shows better skill, basically because it allows us to better match the vertical mixing patterns along the seasonal cycle in float observations. And we are also minimizing the spatial mismatch that happens when you compare a 3D simulation and real-world observations, because the 3D model has its own dynamics and sometimes it doesn't reproduce the features at the right place or the right time, right? So, well, there is a lot more to this comparison, which I can talk about, and it's also described in the paper. So basically, for us, this tells about the two types of error that are influencing this comparison is the sampling and representativeness error, and also the error in forcings and boundary conditions. But these are not the ones I'm most interested in, but I'm more interested in these other types of uncertainties, so parametric errors in the model, and also the importance of model equations and potentially missing processes in model equations. So to do that, I decided to to do some model parameter optimization to try to make some sense of observations and models together. So as you can see in this time series that I already showed before, when you take the ratio between BGC Argo and the model, you see this mismatch pattern, like this sinking plume of a small POC in the model, which does not exist in, in the observations. And this mismatch is very common to all high latitude floats in the North Atlantic, and to some extent also in the subantarctic regions. So we tried to understand why this was happening using parameter optimization. But okay, so before we did the parameter optimization in the 1D framework, we decided to do some sensitivity analysis to know which of the many tens of parameters that the model has were influencing the most the simulated POC concentrations. So basically, you see here this schematic of the model with several processes influencing the detrital POC dynamics. And I have to clarify that in the mesopelagic layer, most of the POC is detrital, unlike in the productive layer where it's also phytoplanktonic and as Camila explained before. So we basically ran several thousands of 1D simulations where we perturbed 9 of the parameters that we found a priori more influential on the POC budgets. And then we computed the sensitivity, both the first order sensitivity, which means changing one parameter at a time, and the total or global sensitivity, which includes interactions when you perturb several parameters, all parameters at the same time, simultaneously. So perhaps not surprisingly, what we got from this exercise is that the sinking speed, the remineralization rate constant here, X3MIP, and the flux feeding, so zooplanktonic interception of the sinking flux and fragmentation, were the most sensitive parameters of the model. So with this information, we then use the genetic algorithm, which we also implemented and described in another paper, to try to optimize these parameters against BGC Argo data. And so here, I'm showing the results of one of the genetic algorithm experiments for just one annual time series of this float in the Labrador Sea in the year 2015. So this work is described here in this Falls et al. 2022 paper. So basically, in a genetic algorithm, you iteratively refine the parameter values by comparing the simulations to a population of simulations against observations, right? And decreasing the cost function. And what you could see in this experiment, for example, is that the model required a slower remineralization, about half of that, of the default, which is this horizontal line. It required also slower sinking, less than 1 meter per day, about 0.5 meters per day. And it required also higher flux feeding and fragmentation, which are linked in the model. But one experiment of this is not enough. So we ran several ensembles of these experiments using data from different floats and different years in the same region. And very interestingly, what we got is summarized here in this in this graph. So on the x-axis, you see the sinking speed parameter, which with its default value, which is 2 meters per day. And on the y-axis, you see the remineralization parameter, which has this default value. And when we look at the population of model solutions that are in best agreement with the observations, not just one, but just the 25% best performing of the ensemble, we find these two clouds of points. So in both cases, the sinking speed is below the default of the model, so suggesting that the tritral POC in this is mostly almost suspended more than slow sinking, right? And you have divergent solutions in the case of the remineralization, which is interesting because 3 of the floats converge to very similar remineralization parameters, but one converged to higher remineralization. And well, this is a bit complex because of the variable reactivity model that we have in PSCAS, and it is too long to explain here. But basically, there is more than one solution that can satisfy observations. And now I warn you, this is a work in progress, so I haven't come up yet with the ideal parameter set for this model. But if we take this, for example, the 1 meter per day sinking speed and half of the default, so we halve both parameters, half of the remineralization as well, and we transfer these parameters to 3D simulations for the Labrador Sea region, this is what we observe. So we see that depending on the parameter perturbations we apply, we have really important changes both in the sinking flux of small particles and in the remineralization rate. So for example, if we have, and well, I have to explain here that I applied one parameter perturbation at a time and also them in both of them in combination, right? So if we slow down the vertical sinking, of course, we have smaller sinking flux and we have more remineralization because we are letting particles stay for longer at the surface and remineralize there. If we have slower remineralizations, we have more particles available for sinking. So this increases the sinking flux and decreases the remineralization in shallow layers, but then it increases it below because there is more particles that make it to deeper layers. When we apply both perturbations in combination, the result is this green line, so a decrease in the sinking flux and a slight decrease in shallow remineralization. And what is interesting here is that this combination is a bit non-linear because if we add the two perturbations of one parameter at a time, which is the gray line, we obtain a result that diverges quite a bit from the simultaneous parameter perturbation. And I'm sorry, I have to leave it here because this is a work in progress. And it's super challenging, I found, to optimize these model parameters. And also there's lots of uncertainties, which I will explain here in the conclusions. So essentially, I have shown that there is a reasonable correspondence between the backscattering measurements and the simulated small and large retractile POC pools in this model, in this case version 2. And I have shown that these 1D simulations are very, very appropriate for this parameter optimization because they are lightweight. They enable like extensive exploration of the parameter space and optimization exercises. I have shown that this, by changing parameters just within plus minus 50% of the default values, which is less than the actual uncertainty in these parameters from the real-world observations, we have a strong impact on gravitational small particle export and generally on POC budgets. Also, there's lots of problems with this approach I took, which I'm happy to recognize because I have been learning through this process. And first of all, I think I was over-optimistic in many aspects because there is still lots of uncertainties in converting backscattering to POC, especially when it comes to seasonal variations and the vertical axis as well. Also, the transfer of parameters from 1D to 3D simulations is not that straightforward. So something that does a good job in 1D may not do a good job in 3D because of the horizontal transports and other things that happen in the 3D simulation. And of course, that using only backscattering, even if we have high resolution observations, is probably not enough to constrain all the fluxes that contribute to this biological carbon pump. So we need to use other observations. But this is controversial. It is complicated a little bit because if you introduce other observations that are not high resolution or are even climatological or do not reflect like episodes like backscattering data do, and we don't know to what extent this will bring new information, but it has to be tried. And that's it. Thank you for listening. I'm happy to take talks, to take questions.
Well, thank you, Martí. It was a really interesting talk. So yeah, we'll open it up to questions. You can type your question in the chat here or raise your hand, and we'll go to you. Ken Johnson.
Hey, great talks, both of you. Here's and especially Martí, but in these comparisons, there's still a huge fundamental uncertainty in the data we're trying to match, which is like POC or the phytoplankton carbon. And I will go back to the point that if you read the literature, we still don't really agree on how to filter POC and whether it's a 5 liters or 1,000 liters, you get fundamentally different results. That's dates back to the Jagoff's days. And I don't know what my question is, but I guess the fact that there really is fundamental uncertainty in how you measure POC, and which all gets back to filtering, which probably that's mostly an issue in the deep water, but maybe not so much for phytoplankton. But I have to guess still that when you're counting, flow cytometry data, whatever, and you need to know, carbon to volume ratios, there have to be. So I guess my point is that, so being a chemist mostly, I kind of like the methods that use oxygen and nitrate and carbon to get at fluxes, and really rather than given the uncertainty in the optical method. So how do you, any sense that, oxygen methods, for example, looking at the respiration in the deep water would be a better way to get it, maybe a way to calibrate these optical measurements?
Okay, I can reply. Thanks for this question and comments. I think I can only agree that there is a huge uncertainty directly in how we measure POP to start with. Well, not huge, but a large uncertainty, especially in deep waters where the concentrations are so small. And I remember that Jim Bishop he reviewed the paper, my 2022 paper, and was asking questions about these deep POC concentrations, and was suggesting us to use data from his measurements with large volume filtration devices and pumps and so on. And in fact, we tried to make these conversion factors consistent with those measurements, but still, I think going to the second part that so biooptics is powerful because of the resolution, the time resolution they allow. But I agree that biooptics alone probably does not give enough constraints to calculate the fluxes or to refine the fluxes in the model. So if I started this today, because this is something I started like several years ago, and as I progressed, I realized that I need more constraints, but at the same time, and oxygen and nitrate are obviously constraints that I would use. And today, it's sad that when running these optimization experiments, it's not possible to go back because you run the experiment with observations that you choose at that moment. But for example, if I did this today, I would go for another approach, just perturbing the many parameters simultaneously, and then using Bayesian techniques, which allow a posteriori to compare to different types of observations in different combinations. And I guess that's answers part of the question.
Thank you.
Do we have any more questions? I have one for Camila. I was just wondering, because you looked at the chlorophyll backscattering relationship, and I was just curious to know how you took into account like the fluorescence to chlorophyll relationship, like what quality control data did you use from Argo, or did you calculate slope factors? Is that something you thought about when you were doing this? So
no, from the Argo, you mean, not direct for the Argo data. I didn't do much. I took the from the MATLAB toolbox all the data, and that's what I did.
I see. Yeah. Okay. Interesting. All right. Just curious. Yeah.
So I have a question for Camila, perhaps.
Oh, go ahead.
Basically, so it's very interesting what the exercise you did, right, to reconstruct from these model simulations the backscattering. And in fact, I think it would be more appropriate to do what I did with a model that simulates the backscattering. But the question is about the detrital fraction of the POC in surface waters, which, as you explained, contributes to this intercept of the relationship. And you explained that it's the majority of the POC stock in so most of the time. And so so it's late here. I'm trying to remember my question. So do you think we could use in combination your approach and the bulk backscattering to POC conversion to try to refine a little bit the comparison against models, for example?
I mean, I'm not sure because that's, yeah, I'm also just using a model basically. So, I mean, especially the POC. So we paid a lot of, well, we paid a lot of care on the parameterization of everything, but precisely the POC, what we do is we actually only have one particulate POC pool. And then what we do to convert it to the backscattering signal is to assume some kind of size spectrum. And then based on some kind of size spectrum that scales with the total POC biomass, we can get the, well, we derive the backscattering of the, like the average backscattering of the total POC, you know. But I mean, I guess we would have to, yeah, I'm not sure I would dare to use it to constrain other models, you know, because this was really, as I say, was a modeling exercise. We compared the model with data, okay, with the model, with the data that we had. But yeah, we are still really, I guess I should have to compare it first, the POC itself, our POC biomass with POC derived from Argo, even given the uncertainties or not, but to be happy and and see that, say that we can use it. But yeah, so right now I wouldn't dare to use it as a constraint for other models. That's the conclusion, basically. Yeah.
Great.
Yeah,
So,
thanks.
oh, sorry, go ahead.
Well, I was curious also about the heterotrophic bacterial backscattering, because you show that in some regions, seasons can contribute quite a bit to the backscattering, and this has not been very widely considered in many studies, right? So
no. So we have. Oh no, sorry. Go on.
Yeah. So, but is there a comparison, like observational estimates of the bacterial contribution to the backscattering that you can compare this to, or?
No, not as far as I know. I guess. No. So that's something that we derived, and actually, I mean, we had the parameterization. We know bacterial size, cell size, and then we had some data. I think it was from the Stramsky 2001 paper of the backscattering associated to bacterial size. But yeah, from there to whether the model simulates reliably the biomass of heterotrophic bacteria, and consequently backscattering might be, yeah, I don't know. We cannot really compare it, you know. Well, I guess, I don't know, is there biomass of heterotrophic bacteria data? I would be happy to look at that. I don't know. Anyway, I haven't done that. So yeah.
But there's lots of flow cytometry data.
Yeah, so it could be
assumptions.
Yeah, so that could be an option, I guess. Yeah. But no, I haven't checked that. Sorry.
Well, very nice study.
Okay. If there's not any more questions, I don't see any in the chat, or no one's raised hands are raised. So we'll just give the speakers one final thank you for their talks and staying up so late. They're in Europe. So thank you again. And thank you, Yui, for the update on the GO-BGC array. And thank you, Heather, for Heather Benway for organizing the Zoom here. So yeah, and if you have any questions, I'm sure, you know, if you reach out to the speakers here, they'll be happy to answer over email or something. So
Thanks, Adam, for hosting this webinar.
Thanks.
Thanks everyone for joining.
Thanks to all.
all right.